Parts can also exert their function on the cell at the DNA level. By this I mean that the molecule that performs specific interactions with other molecules in the cell is the DNA itself, not an RNA or protein that it encodes. One category of DNA parts are promoters. Promoters are defined by their propensity to initiate formation of a new RNA molecule in transcription. Like protein coding parts, they can be functionally or mechanistically characterized. The simplest are the constitutive promoters. These are sequences that directly bind to sigma factors and initiate transcription. They do not require or respond to additional proteins like transcription factors. All other promoters are some type of conditional promoter in the sense that they require something other than just sigma factor for function. TF-dependent promoters require a transcription factor. Some promoters use a different polymerase of viral origin to produce RNA. These include the T7 and sp6 promoters, which require T7 RNA polymerase and sp6 RNA polymerase, respectively. Others employ a normal sigma factor-dependent promoter, but also a transcription factor. SPO2G promoter of B subtilis is one of these, and it requires a transcriptional activator called SPOA for function. Others bind to and are repressed by a transcription factor. TF-dependent promoters can also be engineered to respond to an engineered transcription factor. Particularly in eukaryotes, a zinc finger protein can be engineered into a transcription factor for arbitrarily chosen sequences. Promoters can also be categorized by their inducibility with an external chemical trigger. For example, the LAC promoter is normally repressed by binding of LAC-I. In the presence of IPTG, LAC-I dissociates from the DNA, resulting in expression. The LUX-I promoter, in contrast, is activated by its ligand-dependent transcription factor. LUX-R only turns on LUX-I when the ligand is present. Other popular inducible promoters are TET repressor and PBAD. Mechanistically, these systems are not similar, but for design, they play a similar phenomenological role in the cell of causing transcription in response to an external chemical. Finally, there are environmental promoters. These are promoters that respond to pleiotropic transcription factors in the cell in response to external stimuli. Examples include responses to iron, oxygen. There are other DNA elements that I'll collectively call cis elements. They cause the larger DNA encoding them to undergo some reaction. Origins of replication are one category of these elements that initiate replication on the encoding DNA. If you'd like to read more about origins of replication and some of the details about their use, check out this URL here in pink. Origins of transfer initiate rolling circle amplification and transfer of the encoding DNA to another cell during conjugation. Origins of packaging initiate the packaging of the encoding DNA into a phage or virus particle. Other DNA sequences are the site of reaction of a DNA modification enzyme. For example, the sequence GGATCC is the part that is acted upon by BAMH1 restriction enzyme. Recombination sites are another type of part that are sites for recognition by sequence-specific recombinases and integrases. We've seen this before with LOX and its recombinase CRE, FRT sites, and Hicks sites. Terminal repeats are DNA sequences that are acted upon by a transposase. They are the sequences bound by the transposase, are cleaved by the enzyme, and become covalently attached to it. This results in a transposome that can go on to randomly integrate into a recipient DNA.